Great. So my second talk, I will focus on gene therapeutics for motor neuron disease because the main focus in my lab is developing and using viral vectors to establish and test therapeutic gene therapy to rescue neurons, in particular motor neurons, in animal models, but also taking this through translational pathway. This is just a summary of some of our activities that I'm doing. I will start with this definition of advanced therapies because it's very important. It's quite a very fast-moving, growing space area. And the term of advanced therapies covers highly specialized medical treatments such as cell and gene therapy. And it's, as I said, it's fast growing, and it's expected that the advanced therapy sector, including gene therapy, will grow to a market value of $14 to $21 billion per year globally by 2025. The main focus on my second presentation is gene therapy, and it's using mainly viral vectors that have been modified to remove any harmful properties and make them very safe to be used into patients. So gene therapy obviously had some hard time, mainly in early 90s, with some bad news, and unfortunately some of the investigators were cutting corners originally in terms of various aspects and not making sure that, for example, manufacturing clinical grade vectors are clean enough and pure enough to be safe and used in patients. And that leads to some patients dying, and made and caused hard time for gene therapy in the 90s. But since then, the field moved really fast, and major advances were made in the last 15 years. So in particular, viral vectors were refined to high efficiency in terms of safety and also efficiency in terms of efficacy in treating both animal models but also patients. Some good news in terms of the first products that were approved both in the US and FDA and European Medicine Agency, in particular AAV9-based gene therapy for spinal muscular atrophy, and other products that are coming up and were approved, received marketing approval, authorization recently. The other encouraging aspect is that regulatory bodies like FDA introducing fast-track approval process to bring these therapies to close to patients as soon as possible. And this obviously helped quite a lot. So as I said, we use viral vectors to introduce therapeutic genes into cells. And these viral carriers are modified to remove all the harmful properties. And the most exciting, or the gold standard vector system currently in use, is adeno-associated virus. And probably among all the participants in this conference, in this school, probably 50 or 60% of us are already carrier or infected with adeno-associated virus, because it exists commonly in nature, and it's not leading to any causing any human disease. It's widely used in clinical trials, and I will show you some of the examples of that later on. So adeno-associated virus, we don't use the wild type virus. we introduce changes. We remove all the viral genes and protein. We make space in the lab to introduce a therapeutic cassette in this vector, in plasmid, and we keep only the key elements that will allow us to package the virus and introduce it, and the capacity to infect cells. You can see that we use other plasmids providing the capsid proteins to make the virus into cells. Adeno-associated virus, some historical data, and pre-2002, you can see here that only a couple of serotypes exist. Depend species from where these viruses are coming from. So you can see that around 6 or 7 serotypes existed before 2002, but post-2002, a large number of other serotypes, AAV serotypes were discovered or were generated in the lab through different processes. And you can see here some of these processes that allow to engineer a rational design of these capsids to pseudotype AAV viruses. So these AAV viruses are as I said, widely used in clinical trials, but research, preclinical, but also clinical applications. You can see here the different routes of administration that are used to deliver AAV. AAV is very good at infecting muscle cells. It's great in some applications in the eye. In the CNS is the golden standard, as I said before, so both for the brain and spinal cord disorders. Other applications, we have a serotype AAV serotype 9 that when introduced in the blood, it can cross BBB, but in particular in young individuals. And it has been used this way in babies and children with spinal muscular atrophy. Just to give you an idea about some of the programs that we are running in Sheffield, we use the AAV to replace genes, the missing genes, introduce neuroprotection or gene silencing, but also gene editing as well, and other applications. We have collaborators, for example, hearing loss and manipulating other therapeutic targets. Why we use adeno-associated virus serotype 9? For the following reason. When we deliver this virus in mice through cisterna magna in the CSF, cerebrospinal fluid, you can see here that this virus expresses GFP protein. It's going everywhere, so you can see the cerebellum and the brainstem, the cortex, and different part of the brain, hippocampus, but also in the spinal cord motor neurons, as you can see here. So it has been also reported that when delivered in patients and newborn animals, but also children before 2 years, through systemic delivery, it can cross BBB and in fact transduce cells in the brain and spinal cord. Obviously, transduction in adults is reduced after systemic delivery. That's the reason we go directly in the CSF. And this is a very good alternative to introduce therapeutic genes in adult patients. I will just start with this first example, which is SMN replacement gene therapy for spinal muscular atrophy. I will not go through the entire pipeline of gene therapeutics we have at Sheffield at the moment. So spinal muscular atrophy is as I said in my first talk, is a childhood form of motor neuron disease. It existed 3 types: type 1 severe cases, type 2 intermediate, and type 3 mild forms. And the severe cases, which represent around 50% of cases, it can lead to death of children before the age of 2 years if they are left without respiratory assistance. And the disease is caused by mutations or deletions in a single gene, a survival motor neuron gene. What this do, it's leading to a depletion of SMN protein and lead to cause significant reduction in the number of motor neurons, and patients die by, in most of the cases, by respiratory failure. So obviously SMN replacement is a very good rationale, and it's a good target, a therapeutic target. And that is what we did with using AAV9 to replace SMN. And we did this first in vitro, but also in the animal model. You can see that this animal model of spinal muscular atrophy, if we leave this animal without any treatment, or we inject just a control virus expressing GFP, this mice die on average 11 days or after birth. But when we administer the therapeutic AAV9 expressing SMN through the facial vein, so in the blood, in postnatal day 1 pup, you can see that we managed to extend in a remarkable way the survival of these animals. And we also did a dose escalation study where this animals survive even longer, and the maximum survival we observed is over 380 days of age in this animal model. So this was a very nice proof of concept, and obviously this strategy was taken forward by Avaxis, which is now Novartis gene therapy. And they used exactly the same strategy, AAV9, delivering SMN protein in babies suffering from spinal muscular atrophy. And what you see here is the outcome of the phase 1 clinical trial using this strategy. Just if you look at this line here, the green line, this is based on the historical data, the historical study. At 20 months of age, you can see that only 8% of these kids, babies, are event-free. Basically, they are not under permanent ventilation in hospital. However, when the patients were treated, the babies were treated with AAV9 SMN, you can see that most of these children go beyond 20 months without any permanent ventilation, and they were free from this permanent ventilation, which is a remarkable outcome of this clinical trial. And again, so the virus here was delivered through a systemic delivery in blood, and now over 600 SMA kids are treated with this therapy, and the product reached the market already to be used in patients. I will just move to the ILS now, and I will focus on gene silencing. And we developed this strategy to silence SOD1 because of the availability of the animal model. And I will not go through again this. I spoke about what ILS and the incidence and genetics. I will go through this. These slides just summarize the 2 approaches that we published already. the first proof of concept. We reported it back in 2005 using the lentiviral vector, but instead of delivering lentiviral vector through blood or in the CSF, we used this lentiviral vector that was pseudotyped with rabies G glycoprotein delivered in muscle, and these viral particles are taken back to spinal cord motor neurons. And the outcome we got at that stage was that time was really remarkable. The only problem with this approach is that targeting every single muscle in patients will need a huge manufacturing effort. You will need liters of virus per patient. The cost of that is significant, but also there is no manufacturing facility that will manufacture that amount of virus to cover all the muscles. So we moved to a second approach, which we based on AAV on this occasion, and we delivered AAV in cystina magna CSF. I will go through some of the data we generated. I explained this in my previous slide using lentiviral vector. And the lentiviral vector gave us very significant knockdown or silencing of human SOD1, as you can see here. When we treated cortical neurons taken from embryos emerging from SOD1 transgenic mice. And this knockdown or silencing, the depletion of SOD1, human SOD1, is also reflected at the level of mRNA here. So this virus lentiviral vector, as I said, was administered in different muscles of the mouse model of ALS, and lead to extension of the delay or delay of the onset of disease, but also extension of survival in this animal model. This was a remarkable effect, actually, but it needed to inject the muscle in leg, the virus in leg muscles, facial muscles, intercostal, diaphragm, tongue, everywhere basically. And the rationale behind that is that the patients also have muscle atrophy and degeneration of motor neurons everywhere in the brain and spinal cord. And you need to target all these motor neurons to get efficacy. So, as I said, this was a great proof of concept, but unfortunately, the quantities of vectors needed to target muscles was phenomenal, and it was impossible to achieve. So we moved to use AAV9 as alternative. So AAV9 was encoding also siRNA targeted against human SOD1 and delivered into CSF, cystina magna. And this is how it looks like when you deliver the virus into cystina magna, basically. And the team followed these animals after delivery of the virus, either at day 0 after birth, or 40. And you can see that Tom and Joe in the team who did these experiments followed these mice using different behavior and motor testing. This is just an idea about illustration of the biodistribution of the virus transduction in the brain and different part of the brain and spinal cord. So when we looked at the impact of the treatment on this, what we call a neuroscoring, and this is what it means. So the onset, abnormal gait, wobble gait, dragging of one hind limb, for example. You can see here that the treated animals with the therapeutic vector virus didn't develop these symptoms or deficits only after 110 or 120 days of age, while the the mice treated with the control virus, they start developing these deficits as early as around 70 days of age. What this means is that the onset of disease was delayed in treated animals compared to controls, but also we looked at the motor deficits in these animals. You can see that the SOD1 transgenic animals, they develop a very sharp decline in their motor function with age, but when treated with the therapeutic virus depleting SOD1, you can see that they have stable motor performance. This lead also to delay or extension of survival. You can see we extended the survival here of the mice by around 62 days compared to control mice. I don't know if I cannot get the video to work. That's not a problem. I will move to the next slide. The impact of the treatment is also very significant in terms of motor neuron protection. So we quantified, the team quantified the number of motor neurons in the lumbar spinal cord of treated mice versus controls. And you can see here using nasal staining or CGRP staining, both this labeling allowed us to illustrate the impact of treatment in protecting motor neurons in treated animals compared to controls. This mice also develop reactive gliosis in the brain, and you can clearly see that here. This is the treated mouse with control virus compared to treated mice with shRNA against SOD1. So clearly, SOD1 depletion leads to a reduction of reactive gliosis here. What is exciting also is that we developed this biomarker that can be used in clinical trials. So we took a CSF, cerebrospinal fluid, from these mice. You can clearly see that the treatment lead to 63% depletion of the level of human SOD1 in CSF in the treated animals with therapeutic vector compared to controls. So basically, this can be translated into a human clinical trial by taking CSF samples pre-infusion of the therapy and post-infusion to demonstrate the therapy and as a readout for the therapeutic efficacy. We also tested the specificity of the sequence, shRNA sequence that we used. You can see here that we tested this in 2 cell lines, a stable SOD1 cell line, but also an astrocyte, astrocyte cells emerging from patients. And both cell lines clearly demonstrated that the shRNA sequence we use don't target any unspecific. So the clear target is SOD1, as you can see here. So this is just a summary to finish. We have AAV-mediated gene transfer, efficient AAV9 mediated gene transfer to CNS, excellent proof of concept, and we are working now with various collaborators to take this forward. And the other exciting thing that this project and package of next steps was discussed with the regulatory body in the UK and gave a very positive feedback on the program. In terms of the relevance, clinical relevance of this strategy, targeting SOD1 as a therapy, my colleagues in Sheffield, clinicians doing clinical trials, and this is a collaboration with Biogen, using antisense to deplete SOD1 protein in patients with ILS mutation, SOD1 mutation. And clearly here, this was reported in the New England Journal of Medicine, showing very, very promising efficacy and safety of this approach by depleting mutant SOD1 in ILS patients. So this is just to thank our collaborators and the funding bodies that allowed us to do the work that I shared with you today. And thank you very much, and happy to take questions.